when I was invited to do the talk at this conference, I thought about what we should cover, and then I came up with the idea to call it simulation of life. Simulation of life, that's a very big title, and it's a vast field that covers a broad variety of different topics, ranging from computational chemistry up to artificial intelligence and human behavior. Instead of giving an overview of all the possible topics that may fall under the title Simulation of Life, I would rather give 4 selected examples out of our current research that all have in common that somehow, one way or the other, deal with living matter and the simulation of living matter. And they do this on several levels of scale. Starting on the atomic level, where we simulate the behavior of atoms in molecules and in a chemical environment. We also visualize this behavior with global rendering techniques. Then we get one step further to the cellular level, where we simulate the behavior of single and multicellular organisms. And the next step will be the sensory level. One very important issue when we're talking about life and living systems is the information exchange with the environment. And this comes through sensors, and the simulation of biological sensors is another topic that we will talk about. And finally, we end up with the simulation of human behavior in a traffic simulation environment. We lost the presentation. Starting with the atomic level, I don't want to explain these equations, but one thing important about computational chemistry is that we are calculating force fields to simulate the forces that are present between the single atoms. It's impossible to solve the Schrödinger equation for every electron in a molecular environment, but there are some simplified approaches to that, and these simplified approaches are called force fields, where we try to somehow simulate which forces are important when atoms come close enough together that something interesting is happening. And one approach to this simulation is the molecular dynamics approach, which simplifies the model of atoms to being rigid bodies. And if they were rigid bodies, it would be possible to solve the Newtonian equations and treat them as we treat macroscopic objects, and just follow the movement that these atoms have and show what happens then. Show the dynamics of these molecular systems. And that is what happens here, and there are some interesting things that can be found out when we apply this molecular dynamics calculations to biological systems or biochemical systems, to be honest. In this case, the biochemical system is a rather easy one that consists of 2 lipid-type molecules. Lipids are molecules that are present in biological membranes, and these are 2 molecules that can be found in biological membranes, or at least can be placed in biological membranes. And here we simulate what happens in such a system, which is one very, very small part out of such a membrane. And if we check the dynamics, the movement of these molecules, then we see that these two types of molecules, these two different molecules out of these membranes, have different behavior. In that left one, here, has more flexibility, while the right one is more stable. You can see this, that the amplitudes are lower. That means that the molecules are more stable. Now, what does that help us in terms of simulating life? We are far from simulating life with just these two molecules, but these molecules are present in plentiful in, for example, bacterial membranes, and this is a part of the membrane of a certain type of bacteria, and this becomes interesting when we are looking for antibiotics. Many antibiotics work in a way that they damage or destroy the cellular walls or the external membranes of bacteria. And this is important because they don't affect human cells or other mammal cells. They only work on bacteria, and this is the reason why we can take this large amount of antibiotics without any risk for ourselves, but the bacteria die. And many of these antibiotics work on the outer membranes, and therefore it's very helpful to know the behavior of these outer membranes when we are designing new antibiotics. Another way to apply antibiotics is to stop the process of protein formation in the ribosomes. What we see here is a part of a ribosome, and the ribosome is the protein factory, we could say, of any cell. But these ribosomes, although they are quite similar in all living cells, still have some differences between bacteria and, for example, mammalian cells. And this here is a bacterial cell, and here we see the antibiotic, which fits very closely into a gap between two subsegments of this bacterial cell. And it stops, or at least slows down the effect of this ribosome, and therefore proteins that the bacterium would need are not generated, or they are generated too slowly, and then this is a problem for the bacterium, but not for the mammal that carries the bacterium. Therefore, it's helpful, and now we can simulate how this antibiotic works. And when we apply this molecular dynamics, we can see that here is this part of the ribosome without the antibiotic, here it is with the antibiotic. And if we look closely, we see, for example, here in this part, that the structure has become more flexible, less rigid, when the antibiotic is bound, while in this part here, the flexibility has been reduced, and therefore we could estimate that this antibiotic works in stabilizing the ribosome, and if it's too stable, then it cannot work. There's no motion, or at least too little motion, and this is the way this antibiotic works. And if we better understand that, then we are in a better position to design new and better antibiotics. Now, we only saw static images now when we are talking about dynamics. When we're talking about dynamics, it would be more helpful to see motion features, and that's what we are trying to do now. In the first video now. when I prepared the presentation, it worked on my laptop to show it in the presentation here, but just in the check right now it didn't work, but we still can show the video, I hope at least. And then we can see in this video that we are able to apply several rendering techniques to a very large system. This is the part of the ribosome we just saw. Every sphere is an atom within that. The red spheres are oxygen atoms, the gray carbon, white hydrogen, and the others are several different atom types. But here we just see testing the environment with several parameters, playing around with parameters. And now here we see some global illumination. We see that shadows are cast, or at least it looks as if shadows were cast, and we get a better impression of the three-dimensional arrangement within this structure here. And this is a real-time rendering scene, so it's not pre-rendered. This happens on the screen if we apply this procedure and this software we wrote. And we see this somehow fuzzy shadows, which is the approach to simplify the rendering procedure. Here we have some focal depth that we can imply on this scenario, and several other features that usually are very cost-intensive when we do this rendering. But in this case, we have applied special algorithms and special ideas to make it possible to actually render this in real time with the best possible resolution regarding the spheres, because these spheres are analytical spheres and not triangle meshes like usually when we have real-time rendering. So now we need the PowerPoint presentation again. Comes back.
Wolfgang, excuse me. And where is your antibiotic in this case?
Oh, the antibiotics?
Yes.
With books here. I just showed it was a small molecule that was in the gap between the RNA and the protein part of the ribosome.
Only without the interaction with antibiotics, yes?
Sorry?
Without antibiotics.
Was just the antibiotic molecule in the heart of the ribosome, without the rest of it. In the dynamics scene, you're asking where it was in the dynamic scene. Actually, I couldn't tell, because this was just taking the system and trying the rendering approaches to the overall system. The video was produced just a few days ago, very quickly, and we haven't optimized it to show the antibiotic. We just want to show the procedure here, and that is possible to have a look at that. If we would want to show the antibiotic, then we would just give it another color, and it would be better. Thank you. So here we have several other scenarios, and we are using some knowledge about human perception and some drawbacks within human perception that we can use to enhance the rendering and to speed it up without human spectators recognizing. And we can see in the next video that we have an entire virus hull. It's only the outer wall, not the interior of the virus, but we can see the entire virus, which is a smaller one, about 5,000 atoms, and the larger one, more than 10,000 atoms, that are all rendered in real time with the global illumination model. And we will see the scene now these two scenes in the next two videos. I think it's impressive just to see how the different types of rendering give a better impression of the spatial arrangement of atoms in the system. This is the smaller virus, it's a foot and mouth disease virus. And here we have the entire hull of this virus on the atomic level. Then we will get to the different rendering part. Now we have a different type of illumination that makes the 3D arrangement better visible. And then ambient occlusion, which is a kind of shadow casting that can be rendered much more quickly than the regular ray tracing procedure. And here we see that we can get as close as we like, and we never see any triangles because the spheres are real spheres that are rendered directly. And now we are inside the virus. Usually the virus is inside of us, and now we just change this in the other way, but we don't damage this virtual virus. Here you can really see quite well that this shadow casting improves the impression of spatial depth within this very large molecular structure. Here again we have this focal depth feature, which also gives a better impression about spatial depth here. If we change the level of focus in the system. Now I think we've seen enough of this video here. And then we can also we can get to the next one. That's an even larger virus, the Zika virus in this case. And in this case, rendering would be even more expensive in terms of CPU power or GPU power, because we have a ball and stick model where we not only show the atoms as spheres, but also the connections between the atoms as cylinders. And if we use triangle meshes for all these bodies, it would be a whole lot of triangles to render, but in this case it's done with a different technique. Now we're getting back to the PowerPoint. Here there are some principles we apply. There is a low latency rendering, which makes rendering much quicker, makes it real-time possible. We use, as I already said, analytical bodies for the spheres instead of the triangle meshes, and we use some ray tracing enhancing techniques like the ambient occlusion instead of real shadow casting. We use area lights to simplify the illumination of the spheres, and thereby we can apply global illumination models, and we saw this depth of field, which makes the focal level change and gives some impression of more reality, because if we focus on something that is far apart, then we don't see any objects that are closer to us really sharp, and this gives an impression of more realism in the rendering. Now, these are the structures we have just seen, and these are the two videos which we showed. And now we are leaving the atomic and molecular level and taking one step back and looking at the cell as a whole. And the type of cells that we chose to simulate are slime molds, which are very, very interesting organisms that are just at the brink between single cell and multi-cell organisms, because they, during their lifetime, they change between living as single cells and living as aggregates of cells with different behavior within this organism, and therefore they can live as single-cell organisms and as multi-cell organisms. And there are many other very interesting features about slime molds, but for this presentation we just focus on this slide here below, which is called Physarum. That's the species, the biological species. And these Physarum slime molds have very interesting abilities in finding ways and optimizing paths within maze or within any environment, and they build nutrition networks, nutrition distribution networks that are as good or sometimes even better than networks created by humans. And therefore, they're really interesting to research upon. And here, the first step, when you place a Physarum slime mold in an environment where there are some nutrition sources, is that it spreads out and connects in nutrition sources. And it does this in a way that in mass theory is known as the Toussaint hierarchy, which is an order of connectivity, of building up connectivity in a network, and the networks that the slime molds produce are exactly following different stations of this Toussaint hierarchy. And one might wonder how the slime molds that have no brain are able to use these graph theoretical algorithms. But there's something even more interesting about these slime molds. When we look at an experiment that has been done with very many scenarios, in this case it's Great Britain, and what the people did was to place oat grains, which are nutrition sources for the slime mold, at places on a map of Great Britain where the big cities are located. And then they put some slime molds on this map, and the slime molds connected the nutrition sources, and thereby they simulate the traffic network of roads between these cities. And one could find out that the network produced by the slime molds in this way after some time is almost exactly the network that actually connects the cities in Great Britain. And almost exactly means that they found out that in terms of dropout tolerance, the slime mold network is even working better than the human network. And this makes it interesting to see how the slime molds do this. And this is what we do. We are observing the slime molds and we try to simulate the behavior of the slime molds, and then we compare what the real slime mold does and what the simulated slime mold does in analogous situations. But in order to do this, of course, we first have to be able to observe the slime mold under natural conditions, and there is one problem to that the slime mold reacts to light. And it reacts to light in a way that we usually don't like, because then it stops its activity and it gives spores, because normally this slime mold lives in the ground, and if it is exposed to daylight, then that means that the slime mold is no longer in its feeding stage, but in the reproduction stage, and this means they build spores and they don't move, and they don't build further networks, which is exactly what we want to observe. So we should not expose the slime molds to daylight, but we need some light to to observe it, and therefore we built a box where we place some red LEDs in the box to make the slime mold visible in red light, which doesn't affect its activity. Now this seems quite simple, but if we take a look in this box here, we have to produce this light that covers all the entire area quite homogeneously, and we also build this sheltered environment for the slime mold that is not disturbed by other environmental conditions. And this slime mold builds these networks within days. And to produce a video out of that, we usually have to take a photo every few minutes. And this over days. Students are not very happy if you tell them over the weekend you have to take a photo every 10 minutes. And therefore we produced some software to direct the camera, and this is done automatically now. And finally we have achieved a stable environment where it takes these photos over days, and then we have the video of the slime mold. Now, there have been various approaches up to now to simulate slime molds because they are so interesting. And these approaches are done on various bases, starting with some numerical simulation that simulates some oscillatory phenomena. These slime molds don't go forward directly, but always in waves. They are going forward in waves and backward and forward again. This is the way how they move and how they discover their environment. The nutrition network within the slime molds has some flux that transports the nutrition from one part of the slime mold body to another, and this is also simulated with a reward cost-based algorithm. Another example is using cellular automata, like the best-known cellular automaton, the Game of Life from John Conway from 1970, I think, is something like that. And there are graph-based approaches using decision trees for the path planning of these slime molds to reproduce this descent hierarchy that I showed you. And there are already some multi-agent approaches, which are quite new, where parts of the slime molds are rebuilt as mobile particles, and in the environment there are some attractant and some repellent conditions. But all of these existing approaches only focus on a particular part of the slime mold behavior, but they do not simulate everything the slime mold does. They only try to reproduce part of what it does in a particular situation. And what we are trying to do is to simulate the complete slime mold as a whole and its behavior, and we focus on a mechanistic simulation rather than mathematical models that reproduce the result of the slime mold behavior, but not the way it comes to this result. And all the research done up to now, in simulation at least, does not deal with the dropout tolerance, which, to my opinion, is the most interesting ability of the slime molds. And this is what we are focusing on now. And we have modified the approach of Jones with some different agent types. And we have a decentralized decision process which takes place in the real slime mold, as it seems, at the node where it's branching, and not at some central part, which is not really there, but in simulations usually we have the central processing unit somehow, and we also use these attractive and repellent stimuli. Now there are different abilities of the slime mold, and we want to do all of them, and these are some images from our first approaches to this topic. Here we have the different slime mold agents. We use 3 types of agents, which is in fact 2 types with one changing type. And one is some kind of pathfinder slime mold that goes outside and looks for food. And then it becomes a basis that takes decisions. And then there are the nutrition transporting slime mold agents. And with this, we are quite successful in reproducing the real behavior. So what we see here is the real situation where the slime mold is on a petri dish with 2 oat grains here in the end parts of this triangular hook. And there is some aluminium foil, which is something the slime molds don't like and which they try to avoid. So they have to find a way here along this hook. And the real slime mold did this. This are some drawings that show from several simulations what the slime molds did, how they found these connections, and these are some simulation results of a similar situation. Now we're leaving the slime molds and get to another level, namely the sensors of, for example, humans. We have these 5 senses, and all of the senses have a sensor. And what we did here is to simulate a virtual tongue. Our colleagues from Aachen produced some electronic tongue devices here that are electrodes placed on a small plate. And these electrodes react to, in this case, heavy metal ions in aqueous solution. And they just give an electrical signal if a particular heavy metal ion is detected. But there is one problem. Because there is cross-sensitivity between these different electrodes. There is an electrode for silver, for example, for silver ions, for copper ions, for lead ions, and the aluminum, the silver electrode also reacts on copper only in different concentrations. And what we see here is that the reactions of the electrodes overlap, and this makes it impossible to detect which one of several different heavy metal ions in different concentrations really is the cause of this signal of the electrode. And this is where something comes in that we also learn from nature. And the basic idea is to take a look at our visual sensors. As most people know, we have 3 different kinds of color sensors in our retina. One with the sensitivity maximum in the blue part of the visual spectrum, one in the green part, and one in the red part. And therefore they are called red, green, and blue sensors. And these are also cross-sensitive, as you can see from these curves here. The curve of the red sensor, for example, the red light sensor covers the entire visible spectrum. The maximum of sensitivity is here in the orange, yellow-orange part. But the other sensors show different reaction to different parts of the spectrum. But we also can see that if, for example, we have this result here, then it's the same as here, and there is this ambiguity in deciding which kind of light, in this case, produce this reaction. And Therefore, we also have this cross sensitivity, but we are able to detect which color we see, and we are able to do this because we have all these three sensors and some procedure to analyze the signals that come from all three sensors. And to understand what happens there, we need a very small excursion into the world of fuzzy sets. Fuzzy sets are a way to explain categorization in a non-rigid way. For example, if we here see these large and small smileys, and we get the task to put some smileys in the category of small and some others in the category of large smileys, then probably you might come out with something like that. But there is an unclear region, because these two smileys are somehow small, but not that small, a little bit large as well. And this is something that is possible to categorize with some more fuzzy categorization that talks about a degree of membership to the category. And then with this degree of membership, then we can somehow categorize all of these smileys, and we can do something similar with the responses of the visible sensors to light of different wavelengths. And now we can categorize this reaction from the red, green, and blue sensor in a fuzzy way that enables us to detect the original color. And if we apply this method to the taste sensor again, then we come up with also some linguistic variable for the response of the sensor to the heavy metal ion concentration. And when we then combine the different sensors that we have, then we come out with some estimation about what signal caused or what concentration or even combination of concentrations of different heavy metal ions caused these signals. And here we have the outcome, and we have some probabilities for different combinations, and the one with the largest probability is the one that we really use. Therefore we can show that this procedure works and is able to detect not only single heavy metal ions, but even unknown concentrations of several metal ions in the natural solution with this kind of virtual time. Now we're coming to the final part of the talk, and we are again taking one step back from the sensors, which are organs in an organism, up to the complete organism, and not only that, but we are also working with cognitive agents in traffic simulation. Now, what is the idea behind this traffic simulation? We have a virtual environment that we use together with a bicycle, a immobilized bicycle, to educate children how to drive with their bicycles in real traffic. And this education, of course, should be done in a quite safe environment, and this is this virtual environment here. But it also should be realistic. It doesn't help if the environment is too safe and nothing really risky from the real world happens in the simulated environment. But how do we simulate risky situations in a virtual environment? Therefore, we need agents. And these agents, in the first approach, may be just rule-based. There are strict traffic rules that we usually obey, and the first approach is to equip these agents with the traffic rules. But here we have some more or less common situation at the crossroad when from all the crossing roads, simultaneously some vehicles arrive, and now in Germany we have the rule right before left. Maybe here in Cyprus it's the other way because here we're driving on the left, but on the continent it's right before left. And here we have this problem that all of these vehicles have some right neighbor, and they should drive only after the right neighbor. But if they do that, nothing happens at all. They will have this deadlock situation, and nothing will happen. If one of these agents, or if they all decide that we have this deadlock and they drive all in the same instant, then they will crash in the center, which should not happen, and which doesn't happen in reality. So the cognitive agents here should be able to first perceive that there are other agents, and where they are. They should detect when there is a deadlock situation like this one here, and of course they should be able to resolve this deadlock, and they should do that in a believable, human-like way. So not just avoid the deadlock situation because every agent knows about the system. And in order to achieve this, we have defined some cognitive agents that have personality profiles. And there are some approaches that we just our model 1, where we have this personality prototypes, and it's quite simple, 3 different types. There is the under control, the over control, and the resilient prototype, by bringing these 5 types of aspects, personality aspects together. And then we realized that we need an enhanced agent that is able to learn from situations and has some memory. And it should have some perception abilities, because it's not realistic if every agent knows the entire system. So they should have some human-like visible abilities, but not more than human. You should not be able to see what's behind you, for example. And this is also something we implemented. And then you have these enhanced agents that are able to perceive other agents, to detect deadlock situations, use personality prototypes to dissolve their deadlock, because what is really happening in true traffic when we have such a situation, then there are some people who tend to drive into those roads, even though there are others present, and these are the under control. They are bolder than the others, and there are also the over control that wait until there's no other agent left, and the resilient one that try to leave the others drive, but that drive themselves if nothing happens. And with this, we get a more realistic traffic simulation. So we're now coming to our conclusion, and what may this be? We can use the simulation of living systems on various levels of scale to first learn about nature, which is the more scientific approach. But we also can learn not only about nature, but also from nature, for example, from the spine molds or from the human visible sensor, to design some engineering, some technology that can help us in our daily life. And this is the end of my talk, but also I have to thank a lot of people. Many people have contributed to the experimental and the research results that I presented, and of course also thanks to you for your interest and attention, and the organizers of the conference for the invitation and all this organization. So, thank you.
Actually, we have not so much time, so I believe one question will be nice and the others will be later. Do you have any questions? Yes, please.
Do you use real images in your visualization? Or only artificial objects?
Yes, just in the traffic simulation we use some real pictures. So we reproduced some real roads because we use this for the traffic education of children in certain cities, and they have to do some bicycle driving license when they leave grammar school, and then they have to drive in a particular environment in this real neighborhood. And there we had taken some photographs and do this as textures on the models where it is driving. But the rest is always virtual.